Greetings, I am Gail, the head of the DGTalAquaLab, and in this video I will present part of the results obtained in the framework of a collaboration project with teams of scientists from France and Taiwan. Together, we investigated the response of fish spatial distribution to fishing. Let me start with the question: what makes a fish population resilient? One of the factors that influences population resilience is its spatial variability. Several studies showed that populations with highly aggregated spatial distribution exhibit less resilience to a changing environment. And in fishes, the spatial variability was shown to be mainly affected by size selective fishing, global warming, and intensive fishing. Looking at the effect of size selective fishing, the size selectivity of many fisheries, particularly when coupled with prolonged overfishing, can alter the age and size structure of marine fish stocks, as older and larger fishes are often subject to more intensive harvesting than their younger counterparts. In turn, the reduced age diversity can alter the spatial variability of fishes. Empirical work has further found that species differ in how their spatial distribution respond to fishing-induced age truncation. Yet, the underlying mechanism remains unclear. A potential answer lies in the fact that conspecific individuals of different ages occupy different habitats. And removing old individuals from a population can lead to spatial aggregation and increased spatial variability. While this idea is conceptually understandable, it is difficult to empirically test the effect on age-specific habitat preference in isolation. First, because we lack this information, and secondly, there is also a lack of accurate information on how different species vary in their age-specific habitat. To gain theoretical understanding of this issue, we developed a model which allows us to explicitly test how age-specific habitat preferences influence the response of population spatial variability to fishing-induced age truncation. In addition to age-specific habitat preferences, the response of spatial variability to fish-driven age truncation may depend on the environment carrying capacity. So we also took this parameter into consideration. Let's have a look at the model we used. We evaluated our model to simulate two patterns. First, it should simulate an imaginary age-structured fish population with nearly constant size over generations, under no fishing condition. And the second pattern is the proper selection of the habitat based on habitat preferences and carrying capacity. Let's have a look at the entities composing the model. The first entity is the cell that represents a discretization of the distribution range of the fish population. Each cell represents a habitat patch in the landscape and is characterized by its intrinsic habitat index and a carrying capacity, which is common to all cells. In addition, for the case presented today, the cells were arranged as a gradient. The second entity of the model is the fish, which is defined by its age and its own habitat preference value, drawn randomly from the distribution corresponding to the age-specific niche curve that the individual belongs to. And we tested two scenarios, one of which is the fish population exhibit a strong age-specific habitat preference, or in other words, a strong ontogenic niche differentiation, and another scenario with a weak age-specific habitat preference, or a weak ontogenic niche differentiation. In terms of scale, the landscape is represented by an 11 by 11 grid cells with no dimension. The simulations run for 15 years, each year being composed of 40 discrete time steps. These settings allows all fish individuals to reach their preferred habitat within a simulated year. Every time step, the fishes are moving, and at the end of the year, on the 14th time step, the individuals reproduce. They test their survival, and if they survive, they grow older. The individuals in the oldest age class die at the end of the year. At every 39th time step, we recorded the total number of animals in each cell and in age class over the entire landscape. The latter allows us to calculate the Shannon index, indicating the age diversity of the world population, with higher values indicating high age diversity and thus weak fishing-induced age truncation. The total numbers of individuals, regardless of age class, in each cell was used to calculate the spatial coefficient of variation. And we focused our analysis on the relation between these two parameters by fitting the regression lines across all 6 fishing scenarios and considering the slope as an indicator of the sensitivity of spatial variation to fishing-induced age truncation, with more negative slope indicating higher sensitivity and less negative slopes indicating lower sensitivity. Let's look at the initialization of the model. In the beginning of the simulation, 900 fish individuals, 100 individuals for each age class, are randomly located in the landscape. And 6 fishing scenarios of different intensities, including a baseline scenario of no fishing, are imposed on elder age classes at the end of each year. We run the simulations on a gradient landscape with 6 levels of carrying capacity. Let's have a look now at an important task performed by the fish. The moving process in our model follows a modified ideal free distribution, which states that individuals are free to move towards a better habitat when there is no restriction in dispersal, nor intra or interspecific competition. We also consider the fact that fish do not always have the capacity to sense environmental gradient. When Translating this behavior into the simulation, the fish randomly choose a neighboring cell among 8 candidates neighboring cells and compare the habitat suitability of that cell with the one in which the individual is. This realized habitat suitability, RCI, of a cell for a fish individual I, is calculated as a habitat selection function modified from the Basson model of McCall of 1990. It is composed of an habitat preference term, the cell habitat index minus the individual preference, and a density dependence term, the total numbers of fish in the cell n divided by the carrying capacity. The individual compares the realized habitat suitability of the randomly selected neighboring cell to that of its current cell. If the realized habitat suitability of the current cell is higher than that of the neighboring cell, the fish remains at the same location at the next time step. Otherwise, the location of the fish is updated to this neighboring cell at the end of the next time step. Moving on to the results obtained with the model. The first result is that regardless of the age-specific habitat preference and the carrying capacity, the relationships between spatial variability and age diversity, in other words, the slopes, are all negatives. In other words, we always have an increase in spatial variability as size-selective fishing decreases age diversity. The second result is that at any given carrying capacity, populations with strong age-specific habitat preference, in blue, have more negative slopes compared to the ones with weak age-specific habitat preference, in yellow. This implies that age truncation contributes more strongly to spatial variability under strong age-specific habitat preference compared to weak preferences. In addition, when carrying capacity increases, the slope becomes more negative for populations with strong age-specific habitat preference. It means that decreasing carrying capacity weakens the response of spatial variability to age truncation for populations with strong habitat preference. In contrast, for populations with weak age-specific habitat preference, there is little change in the slope with respect to carrying capacity. Together, decreasing carrying capacity reduces the effect of age-specific habitat preference on the response of spatial variability to age truncation. In summary, using an individual-based model, we showed that the interplay between age-specific habitat preference and environmental carrying capacity determines the sensitivity of spatial variability to age truncation. There are 3 key findings out of the theoretical investigation presented here. First, age truncation generally elevates spatial variability. Second, the response of spatial variability to age truncation is higher for populations with stronger age-specific habitat preference, regardless of carrying capacity. Third, carrying capacity alter fish habitat selection process, which in turn influence the effects of age-specific habitat preference on spatial variability under fishing. So, going back to our first question, what makes a fish population resilient, which is important for fisheries management? The present study showed that it is essential to have empirical and theoretical development in a methodology to quantify age-specific habitat preference of marine fish and knowledge on how climatic changes influence carrying capacity. Only with such a knowledge can we make one step closer to even better sustainable fisheries management, which will allow future generations to keep on eating and observing amazing fish from the ocean. The work presented here is from a paper that was published early 2021 in Ecology and Evolution. The link to the paper is provided in the description below. For more information about the research conducted by the DGTalAquaLab, please have a look at our website. And for more videos, don't forget to subscribe. Cheers.